Welcome back, Year 12s, Mr. D here. We're going to go through our next video looking at transcription specifically. So a quick recap of our last video. We looked at some different specific words that are used. Okay, so universal, any living organism, essentially has the same codons which make the same amino acids. Okay, so everything uses the same amino acids. Unambiguous, a RNA codon or a DNA triplet will always code for some specific amino acid. Okay, in this case it's only one. Degenerate, that one amino acid can be coded for by multiple codons. All right, so UUA will always be leucine. Okay, but leucine may also be coded by UUG, etc. And lastly, non-overlapping, they're just read 3 at a time when they go through the ribosome. Okay. Boom. Codon on a triplet chart, you should be able to tell me now. Okay. You see the U's, that is the dead giveaway. Okay, so it's a codon chart. And we looked at eukaryotic genes, how they have a promoter region. Okay, they have exons which are expressed, introns which are going to be cut out, and then a termination sequence at the end there with your stop codons. Awesome. So just keeping in mind, any activities I'll just be skipping because obviously this is just the online video. Cool, so we're looking at transcription today. It's the first of the 3 steps of which a protein or a polypeptide is made. Okay, so transcription, RNA processing, translation. We'll see that in the video today. Cool, moving on to the next dot point as well as still the first 2. Awesome. So now we're moving on to prokaryotic organisms. So, your bacteria, archaea, etc. And as you can see, that the gene structure is slightly different here, but it's made up of similar things. So something to note is that the DNA is much shorter, okay, because generally they're much smaller prokaryotes. All right, think of a bacterium; it's extremely small. All right, prokaryotic genes often contain the promoter region as well. All right, terminator region. What can we see here? There's no introns, okay, but there is exons. And then there's this new region here, the operator region, which in this picture it has a protein attached to it, but we don't need to worry about that yet. That'll be more when we look at the trip operon, okay, but it has this operator region. Cool. So as I said, there's 3 stages of gene expression. All right. Ironically, we just looked at prokaryotes, but these 3 stages are just in eukaryotes because prokaryotes essentially skip the RNA processing. All right. So they just have transcription and translation, which occur simultaneously. Whereas when we're looking at eukaryotes, so animals, plants, etc., all right, there's going to be 3 stages. So transcription, which involves the copying of our DNA. And it's copied into what? What have we learned about last video? Yes. All right. So it's going to be RNA, but in this case, it's messenger RNA. And it's also before RNA processing occurs. So we call it pre-messenger RNA. Okay, as opposed to what happens after RNA processing, which is mature messenger RNA, or just mRNA. Okay, so that's the first step, transcription. When you transcribe something, you're rewriting it. All right, so the DNA is rewritten into pre-messenger RNA. RNA processing is the second step here. Modifies the pre-messenger RNA molecule to produce mature messenger RNA. So things get attached and things happen, and then it becomes mature. Okay, and really this step is just to allow it to go through the ribosome efficiently. Okay, and lastly, it's then sent to the ribosome, and that's where translation, the 3rd step, will occur. Okay. And this step of translation is really just the ribosome going to work, and it's reading the mRNA, reading each of the codons, bringing the amino acids, chaining them together to make your polypeptide. All right, that's the big picture of, well, this video, the next video, and the following video too. So the next 3 videos, we're going to go through each of these extensively. So today we're really just going to look at transcription. Okay. And there's a few terms you want to be well aware of in transcription. Okay, and hopefully you know some of them already, but some of them are going to be a bit new. So the codon, obviously that's the 3 bases when we're looking at RNA. Okay. We're going to refer to the DNA here as codons on either side, though, in this case. All right, we have the coding strand, unzips to allow mRNA to connect to the template strand. So that's that bottom one here. All right, so 5' to 3' end. Complementary strand, coding strand. That's the code of the mRNA that's going to be created. All right, and it's based on this non-coding strand here. Okay, we have the anticodon, which is just the opposite of the codon. So if you see the codon here, C, T, and G, it's going to be the complementary base pairs. So with C, it's going to be G. With T, it's going to be A. Without G, there's going to be C. All right, so the codon is CTG. The anticodon here is GAC. That anticodon is found on the non-coding strand, which is like a template. That's the template for what the pre-messenger RNA is going to be made out of. Okay, so it's a template, it comes in and gets connected to, and then the coding strand is coded. Okay, that can be a bit confusing and students get confused with that. So you may want to re-listen to this part. All right, we also have this enzyme here, RNA polymerase, and as you can see, a quaternary structure enzyme, so that means it's got multiple polypeptides together. So it could be 4, it could be 2, all right, it could be 3. And that runs through it and unzips your DNA, okay? And then it starts to create the pre-messenger RNA. So you can see our strand here. So if we look at this, it's our DNA strand. It's running along the template strand here. And it splits off the coding strand and it will create a copy of that coding strand. Now, if that's a copy of the coding strand, why is it different? Why is this a CUG instead of a CTG? Should be able to answer this by now. Yep, that's right, because this is a part of the DNA, this coding strand, whereas the middle is that brand new pre-messenger RNA that's being made. So all the T's are now replaced with U's. Okay, and you can see that here too. Awesome, and then lastly, we have helicase as well, which disrupts the hydrogen bonds between the nitrogenous base pairs. That essentially runs through with the RNA polymerase. Okay, and again, it ends with A, so it's another enzyme, but it's not shown in this image. Cool. We're going to look at the specific steps of transcription. Again, this is another 3-step process, so not to be confused with the 3 steps of gene expression that we looked at. Okay. So we have transcription factors that bind to the promoter region to initiate transcription. So cool. Transcription factors comes in, and then RNA polymerase comes in too. Okay, binds the promoter region. Weak hydrogen bonds, so H bonds, they're weak. Hydrogen bonds are weak. Between the nitrogenous bases are broken by helicase essentially, and then this yellow thing here, the RNA polymerase will run down the strand and we'll do exactly what we saw in the previous slide. Okay, so that's our first step, initiation. And then we have elongation, which I nearly just said. Okay. RNA polymerase moves across or moves along the template strand. So template strand up the top here. And it's moving across and it's copying that coding strand on the bottom. So you can see that. Oh, well, really, that's actually flipped the other way. But anyway, it's copying the coding strand. Okay. And that's going to make your pre-messenger RNA. So that's elongation. And then termination occurs. Essentially, it gets to the termination sequence, and really it hops off. Okay, as you can see, the RNA polymerase goes whoop and jumps off, and there you go. You have your pre-messenger RNA. Okay, from 5' to 3' end. That will go on to the next step of the entire gene expression process, which we should be aware of. That is RNA processing. Okay. Yeah, and that's it for today. So, yeah, you can watch this video if you like as well. Thank you very much. Have a nice day. Like, subscribe, and comment.